Hello students, I am Dr.R.P.Oviya from the Department of Biotechnology, School of Bio and Chemical Engineering from Sathyabama Institute of Science and Technology, deemed university. I am here to talk about some of the important techniques that are used in genetic engineering. In our previous video, we saw about the overview of what is a cloning method and how a clone is being developed and what are all the vectors that are used for. So here we will see some of the important techniques. Here I am going to show you about SDS page, that is sodium dodecyl sulfate polyacrylamide gel electrophoresis. So in electrophoresis, it's a method where we pass electricity and based on the molecular weight there is movement of molecules. Here it is based on both the molecular weight and the charge of the protein. Here we can see mobility of these protein molecules in a gel. So this gel is a acrylamide gel and the tank it is used for SDS page electrophoresis. So here we use sodium dodecyl sulfate and it plays a very major role. Like a protein is present in the sample, the protein content has to be linearized, only then it is being able to move in the gel. So sodium dodecyl sulfate is a strong detergent and this will help in ensuring that the protein becomes linearized. So once the protein is linearized, the sample is prepared. Like post addition of SDS, the sample is also heated in order to get a linear protein. Then we have to prepare the gel. In the polyacrylamide gel, we can see there is two types of gels, that is resolving gel and stacking gel. In resolving gel, we can see pH 8.8 with 1.5 molar tris chloride and pH 8.8. Whereas in stacking gel, the pH is 6.8, a little lesser, and there is lower concentration of tris HCl. Then there is glycine. The running gel buffer contains glycine and glycine plays a major role. It's an amino acid and this amino acid contains both partial positive and negative charge, and during the separation it helps in the stacking. It ensures that the proteins are being stacked in the stacking gel and then it helps in the movement of the protein inside the gel. So we can see here when the sample is loaded, the sample is heated in along with SDS, it is heated at 100 degrees Celsius for 10 minutes and then it is added along with the bromophenol blue dye. So this dye helps in looking at the movement of the or the mobility of the sample in the gel. So here we can see the in the stacking phase, the molecular weights of the proteins get stacked, that each proteins get stacked along with the glycine, and you can see there is potential difference that helps in concentrating all the proteins in this area. Then once it enters the stacking gel, it goes to the separating where the migration of the protein is present. Here the migration of the protein is completely dependent on the molecular weight, and based on the molecular weight and the negative charge, it keeps moving along the gel. So here we can see that the mobility is based on the percentage of the gel. If the percentage is very low, then the lower molecular weight protein can easily move faster. And hence when we want to resolve a low molecular weight, we use higher percentage of cell for say 12% or 14% of gel. So what are all the reagents that are used in SDS page? We have 30% acrylamide solution, stacking gel buffer and running gel buffer. And in order to polymerize the gel, the important agent includes ammonium persulfate and TEMID. Ammonium persulfate must be freshly prepared every time and TEMID can be used in an undiluted form. So once we have to wipe the glass plates very well with water and then a solvent and ensure that there is no particles present in the gel, and hence wipe it very well with the glass, and then you have to adjust in the frame such that there is no leakage. To test the leakage, we can add water, we can see whether there is leakage present or not. So once we check for the leakage, we can set up the casting gel tray. The casting gel tray can be added and the washing can be done. Initially we have to wash the surface, wash the entire apparatus, and then we have to pour the TEMID. When we have prepared the separating gel or stacking gel buffer, we have to add TEMID at the end. Once we add APS and then we have to add the undiluted TEMID at the last, and then we have to ensure it is just mixed 1 or 2 times and then poured onto the apparatus. So we can see that the separating gel can be poured 3/4 of the glass plate, and as a few amounts of isopropanol or ethanol can be added to the surface so that the gel can be formed in a straight line, so that there is no breakage or there is no bumps in the separating gel. Then the remaining water can be removed out. once the polymerization is done. The polymerization takes even 20 to 30 minutes. Then once it is done, we can pour the stacking gel. Once we have poured the stacking gel buffer, we need to place the comb. The comb, it can be a 12 well comb or a 10 well comb depending on the apparatus. Once we place the comb and let it set for around 10 to 15 minutes, then it is allowed to polymerize. Since the stacking gel is a very low percentage, like 4 to 5% of gel is being used, it is very delicate. So in order to remove the comb from the gel, we have to ensure there is no bubbles or there is no breakage in the gel. Then we have to remove the casting frame, we have to remove the tray, casting frame, and then place it onto the electrophoresis apparatus. Once we pour it into it, we can add the running gel buffer. The running gel buffer contains glycine, and this glycine helps in the mobility of these proteins along the gel. Then we have to load the samples. The sample is in blue in color due to the bromophenol blue tracking dye. So this tracking dye is used in looking for the mobility. Once we have placed the gel and loaded the sample, we have to pass the electric current. The electric current must be passed from positive charge to negative charge. Since the proteins are now negative charge due to the addition of SDS, now we have to let the movement from positive to negative. So we can see the movement of the tracking gel when the electric volt is applied. The voltage can be from 100 to 150 volts, but generally 100 volts will give you a very good fine bands, whereas 150 volts can lead to some disruption in the movement, and hence 100 volts can be most appropriate one. So we have run the gel, we can see the end of the bromophenol blue. The tracking dye has gone to the ends, so the gel apparatus has been removed now. Now once you remove it, do not touch with the hands and ensure that it is being cleanly placed in the beaker. Now we can see here it is incubated with Kumasi brilliant blue. Now to visualize how the proteins are, there are many staining methods. Example, Kumasi blue stain, silver stain, and here I am showing you an example of Kumasi brilliant blue, that is G250. So here the gel is incubated with the G250 dye. The dye solution contains Kumasi G250 and methanol and water. Methanol ensures the gel is fixed and then the proteins does not move out of the gel, and the G50 is stained along the proteins. To remove the excess stain, it is destaining solution is used and the excess stain is removed to obtain fine bands. So based on the molecular weight, we can see continuous presence of these protein bands along the gel. So here we can see that higher the molecular weight, it is near the gel, whereas the lower molecular weight goes to the end of the gel. In the next video, we will see how we can transfer it into the Western blot apparatus. Thank you.